Thank you very much.
Now I'm coming to another company which is probably well known to a number of people, which is Integrated DNA Technologies. IDT was founded in 1987, so it's quite a while ago. They are based in Carroll, Iowa, funded by Joseph Walder, and now it's getting more complicated because I need to give you an introduction of the presenter, and hopefully I'm not misspelling you. Xiangyu Rao. How do you name yourself?
Xiangyu, yeah, Xiangyu Rao, yeah.
That makes it easier. I need next time to write that in phonetically, so that I pronounce it correctly. Anyhow, this company is widely known, and I'm looking forward to listen to your presentation. Thanks. Go ahead, please.
Yeah, thank you, Udo. So, good afternoon, everybody. This is Xiangyu Rao, Territory Sales Manager, NGS from IDT. So sorry that originally I was not the planned speaker because of the Peter Bahasad, our EMEA Field Application Manager, NGS has some private reasons. So that's why I came here just to replace him. So today I will talk about the reliable detection of low frequency variants in highly degraded DNA samples using our Exchange Prism library prep kits. Okay, so first I will shortly introduce IDT, then I will talk about the challenges of FFP samples for NGS. Then I will introduce IDT's Exchange Prism DNA library prep, and finally I will give kind of example to show that how the Prism kits together with our Exon V2 panel, how it works. Yeah. So IDT is one of the biggest oligo supplier worldwide. So it was founded in 1987. The headquarters in Coralville, in Iowa, in United States. So we have 4 manufacturing sites worldwide. 2 of them are in United States. 1 is in Belgium, in Leuven, which is also our European headquarters, and another one is in Singapore, in Asia. So since 2018, we were acquired by corporate Danaher. So now we're a Danaher company. So besides the oligos, we also entered a different application field, for example, qPCR, CRISPR, NGS, synthetic biology. So today I will only focus on the NGS. And also, yeah, this is the high level of NGS workflow. So you could see that IDT NGS solutions covers 3 parts. One is the library prep. So we have Prism library prep kits specifically for FP and cfDNA. We also have Lotus library prep kits for genomic DNA library prep. Besides that, we also focus on the target enrichment, for example, capture solution. Yeah, so as mentioned by previous speaker, currently we also collaborate with 10x Genomics for capture solutions as well. And the last but not least, we also have endpoint solutions named RAMseq. So everybody knows that FFP could be challenged for NGS because of its poor quality and its highly degraded DNA. The fixation could also give some artifacts, and also it's not easy to do the quality control. And also many FFP samples are used for tumor diagnostics or tumor researches for solid tumors. So that's why amount of the tumor DNA in such samples is also very low. So that all the factors could make the trouble to process or to sequence such samples. So so here I would like to how to illustrate here that why the conversion rate in the library prep step is very important. For example, if you have the DNA fragments from your FFP samples for tumor, you see that the tumor DNA in such sample is very, very low. And if you have the high conversion rate kit, library prep kits, then you know, when you are doing the library prep, the complexity of the library is higher, so you have higher chance to engage those tumor DNA during the library prep. And then when you're running, you know, the high deep sequencing strategy afterward, then you will find the signal you are looking for example, somatic mutations or whatever. However, if you are using a low conversion rate kit and with the lower library complexity, then in the library prep stage, you might already lose or missed those tumor DNAs, so which might cause the issue. So you might not able to find the signals afterward. So that's why IDT launched XGen Prism library prep kits March this year, which is specifically for FFP and cfDNA for low frequency mutation detection purpose. So this is the simple workflow of such library prep kits. So you can see that the DNA fragment will be fragmented into small fragments using mechanical fragmentation method like covariates. And then there's no A tiling, afterward. It's not a TA ligation. It's a blunt end, the fragments. And then it's followed by 2 ligation steps using our specific method. So first, the semi-complete adapters, UMI adapters. UMI stands for unique molecular identifier adapters, will be ligated to the blunt end insert fragments. Be aware that this is single-strand ligation because of the 3' blocking group. The ligation of the 3' end of the of one strand of the DNA will not happen. So then, in a second step, there is a single-strand primer will be added to place the small fragment here, and then moving forward to fill in the gap and cross the UMI, followed by 5' ligation. So that's after 2 steps, then you finish the ligation. So by which you already engage those unique molecular identifier besides your insert sites. And in the final step, of course, you need to add the the sample indexes. In this case, we're using unique new index by PCR. At the same time, you also amplify the fragments in your library to the certain amount, which is enough for sequencing. So there are 2 main advantages for such kind of the workflow. The first one is that because of the specific design of your semi-complete adapter, so the chance to have the primer dimers is quite low because of these 3' blocking group, and also the chance to have the cameras is also much lower than normal. And the second advantage is that such kind of the library after library prep contains double-strand UMI, unique molecular identifier there. So which is very helpful if you're going to do the low frequency mutation detection, because you could use those UMIs to do the consensus calling of all the reads coming from the same DNA fragments to eliminate those false positive errors, for example, PCR errors or sequencing errors. So here we did some internal testing using first the FFP 25 nanograms with different kind of quality, high, medium, and low, using the DIN value. We also tested other 2 library prep kits from other 2 vendors, K and N, to do the comparison. So you could see that in such kind of comparison, the yield of the library is higher from the Prism, but also the library diversity or complexity measured by unique molecules in the library is also higher from the Prism library comparing to other vendors. So next, we also run a experiment using Prism library prep kits together with our exome V2 capture panel for the exome sequencing. So in this case, we're using 2 types of the input. One is the FFP samples, another one is the fresh frozen. So we're using 100 nanogram as input. So you could see that the on-type in this case we're using 2 × 10^5 base pair sequencing strategy. I think for each samples we are using 15 million reads per sample. So you can see that on-type rate is quite good, above 95% all the time, and also we are using the percentage of the bases with the 0.2 to 1.5 times mean coverage to measure the uniformity. So you can see that majority of the target are dropped in this kind of range for the coverage, both for FFP and fresh frozen samples. And also another way is to check uniformity of coverage is that we see that majority of the samples have more than 19% of the bases with more than 20x coverage. But last but not least, the complexity is also quite high. So this is evidence showing that the combination of these two technologies, extreme, Prism library prep and also Exome V2, could give quite convincing result, even using the FFP as an input. Although normally people use genomic DNA, but sometimes we do see that the big panels like exome could be used for tumor diagnostic or tumor research. That's why we made such kind of experiments. So next, we're also using a 100 nanogram on-core-span FFP DNA, HD832, to check or to validate the markers like single nucleotide variants or small indels using the Prism and Exome V2. So you can see that from table A, the sensitivity of such panel combination is quite high for all the known markers. Even for the markers with the allele frequency between 0.1% and 0.25%. From table B, we could also see that the specificity is also very high, which means that there's very little false positive SNPs because of this UMI barcode. So because I already mentioned the Exome V2, so I just give a bit information of our Exome V2 panel. So we launched this panel early this year. We designed the probes against HG138 reference, human reference, and using the RefSeq 109 annotation to annotate the exome region. It contains 20K coding genes, and so the footprint of such kind of the panel is 34.25 megabytes. So totally there are more than 400,000 probes covering 39 megabytes of the probe spaces. So we manufactured each panel under the strict ISO 13485 standards, and all are manufactured under the GMP. So because IDT has the specific manufacturing for each probes, that's why the Exome V2 panel has higher uniformity and higher on-target rate. So when you convert such kind of the advantages to the data, which means that if you want to reach the certain mean coverage, amount of the data per sample needed is less than the other vendors. For example, here we tested IDT Exome V2 panel with other vendors, and we can see that you can put 15.6 samples into NovaSeq S2 flow cell if you want to reach the coverage, mean coverage like 18. So that is the advantage for help you to save the the sequencing cost per sample. Okay, so this is the last slide to summarize the advantage for Prism. So the Prism library prep is specifically designed for the challenging samples such as cfDNA and FFP with highly degraded samples. So it has very high conversion rate compared to tail ligation-based methods. Because of the novel ligase and highly modified adapters, it can detect variant at lower than 1% variant allele frequency because of the high conversion rate. Because of the UMI, double-strand UMI, that's why the result not only have high sensitivity but also have high specificities, which means that it can eliminate those possible false positive signals as well. Yeah, thank you for your time, and any questions? Thanks.
Okay, Okay, thanks again for the lively presentation. And we do have actually a question. You mentioned that you can deal also with highly degraded samples. At which time is a sample defined as highly degraded? Can you comment on this?
Okay, yeah. So I think here they're using the DIN value. So DIN value is the value to normally, it's just 2 pairs of the PCR to amplify both long fragments and short fragments. So the ratio, DIN value is kind of the ratio between long fragments and short fragments. So this is the important, because the ratio, when the ratio is lower, then it means that there are more small fragments. So that's why the quality will be worse. Yeah. But there are different ways, to define such kind of quality. Yeah.
Okay, and I see on the same slide, this is probably answering the second question I have. What is the minimum input DNA requirement?
Yeah, so it's a very good question. So, for the Prism library prep kits itself, the range of the, the input DNA is quite wide. So it could be between, 1 nanogram to 250 nanograms. But of course, for the better quality FFP, we suggest at least 25 nanograms. Yeah.
Okay, Xiangyu, thanks a lot. So we are coming now from a long-time player, IDT.